Today, I'm going to talk about this cell tumbling, enhanced stem cell differentiation in hydrogel, nuclear mechanotransduction in Nature Material, published in 2024. Mm. And then this group is from Stanford. Yeah, similar concept with Ovid Chaudhary in Stanford, but they are using different hydrogel. So let's read this abstract first. Cells can deform their local niche in three dimensions via whole cell movements such as spreading, migration, or volume. This behavior occurring over hours today influence long-term cell fate, including differentiation. Here we report the whole cell movement that occur in at the minute time scale, termed cell tumbling, characterized by three-dimensional cell dynamics and hydrogen deformation, elicited by the heightened second-minute scale skeleton and nuclear activity. Study inhibiting or promoting the cell tumbling of mesenchymal stem cells showed that this behavior enhances the chondrocyte. Further, it is associated with the decreased global chromatin accessibility, which is required for enhanced differentiation. Cell tumbling also occurs during other lineages and its differentiation enhancing effects are validated in various hydrogel platforms. Our established that cell tumbling is an additional regulator of differentiation mediated by rapid niche deformation and nuclear mechanotransduction. Here, you have to focus on normal people show us some behavior change over hours to days. But here, they focus on time, minute, second, or this they want. Okay. So they are making two different hydrogel, which is called sliding gel, and then chemically crossing gel. Sliding gel, they have this kind of ring structure. So even though they have covalent cross link, the cell can move the hydrogel locally. But this control hydrogel, they don't have this kind of ring structure, so they maintain their they cannot be easily pulled or pushed by the cell. And then first they culture MSC, two different hydrogel. Actually, these hydrogel also have RGD structure inside. Okay, so cell can attach and then pull and push ECM, and then they culture them MSC and differentiate them 21 days. That they find out this is chondrogenesis staining result. SG has highly differentiated in the chondrocyte, but CG they didn't do that much. But the beauty of the study is that these two different hydrogens, CG and SG, has similar elastic modulus, similar stiffness around like they said 4 kilopascal. Yeah, this is 4 kilopascal around this 3 or 4 kilopascal. So this, spark microbiology, you can imagine you just put the gel in the between plate, then you share it. Using this share, you can change the frequency means how you fast to rotate this plate while you are pushing this hydrogel. So depending on increase of frequency, this is called frequency sweep. Sweep means just do it 0.01, 0.1, 110. Yeah, they increase the frequency, but they maintain this hydrogel stiffness. But when they look at microbiology, they are using the special device, which is called uh, DLS, associated rheology. So the whole idea is that you put some 500 nanometer bead inside of hydrogel, and then you visualize this hydrogel while they're doing rheology thing, and then they track how the bead is moved. Based on their movement, they can measure this kind of complex modulus over frequency sweep. And then they found out that when you look at this blue and blue, red is higher than blue. So what does it mean? This means that this CG has, they describe here well, Sorry. Here. Okay. 1CAB. Here. Mm. The cross-link in both SG are covalent and irreversible, and both hydrogen elastic behavior in cell-relevant timescale, as shown by Burke shear rheology. So from this Burke shear rheology, actually, when you imagine this is viscoelastic gel like alginate, collagen, hydrogel structure, over time, over frequency sweep, this elasticity should be going down. But when they maintain this one over frequency sweep, which means this is total elastic. Elastic means like rubber, right? When you stretch the rubber and then they try to maintain the resistance force, right? But when you imagine the collagen, when you push or stretch the collagen, their stress to resist this, your stretch is going down. This is the meaning of viscoelasticity. So this right figure show us these two hydrogel, they are covalent crosslink, 공유결, and then they have elastic behavior. When they have viscoelastic behavior over frequency, they should go down. Okay, so I am showing viscoelastic and elastic behavior, and then although this demonstrate comparable work mechanical property, we hypothesize that local mechanics to be different, given their like link structure. So to test this, we characterize local mechanics of these two hydrogel using dynamic light scattering based microbiology, adding nanobeads, encapsulating gel, and then their thermal movement over time were monitored to determine the local rheology property. The beads in SG, which has link structure, so they can be exhibited substantially higher mean square displacement at the intermediate cell relevant timescale. He suggests that SG was 5 times softer than CG and local length scale, allowing cells to deform the local niche more easily with the same force. Again, here, when you look at the CG, it doesn't have this link structure. They don't allow sliding. They have higher microbiology value compared to blue. So they said this low scale. So this is like 5 times difference. So it's not easy to understand this, but let's imagine. When you think about the bulk hydrogel, you just bulkly like push and pull or shear it using rheology. They have similar, comparable stiffness, like 4 Pascal. But when you look, they want to know how does inside of local deformation happen. So for measuring that, they're using special device, DLS technology. They are embedded the nanobead, and then they track the nanobead while they are doing this frequency. Actually, I don't know how they induce the summer behaviors. Probably I just imagine this frequency sweep could be a one summer behavior because this is like one way to induce some heat to the your structure. So CG has higher value and SG has lower value, means that while they are rotate this plate and then hydrogel can observe some energy. So while they observe energy, the nanobit can move more in blue one. But the red one, they didn't move that much. This is so one way to show their rationale. So actually, this is this very new concept hydrogel in this field. And then they want to actually, and they want to know why. Okay, actually, they confirm. Okay, nanoscale or over time, over minute, not hours or days, over minute, something changed inside of hydrogel, and then they want to track it lively. SG sliding gel. When you look at cell MSC, see they are rotate, right? But CG, they didn't move that much. So over time, 0 to 16 hour, they track how they move. CG, they almost maintain. Because this is elastic gel, so the stress is not relaxing. So cell cannot move freely. But in SG, even though this elastic gel, they can be slide. So they can move a little bit. Very heterogeneous. And that is their quantification cell dynamics. Over time, this dynamics is go down, but absolutely all the time, as yeah, cell dynamics is SG is always like this compared to CG. So when you look at the ECM deformation, so you stain the gel with red color, and then you track it how this red color ECM they are changed or not. In CG, you cannot see any like big difference, but in SG, somehow this is dance here, dim, dance dim, something happened, right? So which means that cell try to pull and push the ECM, and then sometimes they concentrate ECM here, and then at the same time this is more less. So this, and then while they're checking this ECM deformation, they're checking central speed. Central means the center of the cell. So this center here, here, here. When you look at the center here, almost same, right? But there's why very low value. But this, when you look at SG, even though there are little changes, but this maybe 5 or 10 times higher than CG. This is from the cell whole point of view. And then major angular speed. Angular speed is like angular speed, right? So, okay, what is the major? This is the major axis, minor axis, right? You can see. So the major axis, minor axis, this is axis, how they move here, here, here. So this major axis left unclockwise, right? Here. And then another unclockwise here. But this one, maybe, let's say this is the major axis, almost similar. Over time, this SG, they rotate, right? The rotation speed is higher SG compared to CG. And then for confirming this kind of SG, CG, their dynamics, here they're using AFM and then measure their nano stiffness. Nano stiffness, SG, is lower than CG, right? As work, they have similar, but in nano scale, they are different. And then when they mix half and half, they are in the between, right? So depending on the indentation force, more strain means they are more softer. So always softer than, and then half and half are in the between. They also check this sensor speed versus major axis speed, same time, right? And then I want you to show you this one. Okay, actually these two should be similar, right? But sometimes if you guys show us your data, okay, previously without inhibitor, just you show us, let's say low, but when you do other inhibitor, other study, but that value is very different from previous one. Who believe your data, right? So in single study, the data should be similar shown. Right? Anyhow, you have to do again this one at the same time, right? But at least blue and red should be similar, 0.1, a little bit. This angular speed also 0.03, 0.03. Tendency, so this tendency, its value should be important, like average or their tendency. While you are in a tendency, you can add another inhibitor, another like activator. Okay. So this figure one is enhanced MSC chondrogenesis is associated with mini-scale cell tumbling in 3 dimensions. Here, I did highlight was they encapsulate the cell in 3D, not 2D. You know, 2D is like you see the cell on the top of gel, whereas your interior. But 3D means that you encapsulate inside of gel, you put the cell, and then you maintain the gel, right? So now people want to see what is the real feature in 3D structure, because our body is 3D. And next, let's look at this figure 2 title. Cell tumbling is associated with enhanced second to minute scale cytoskeletal and nuclear dynamics MSC. Imagine, okay, you got this data. Maybe first week, you're making this gel. In second week, okay, cell is trying to move. And then they'll call it tumbling. You know, tumbling is like up tumbling, tumbling. There's a tumbling. So it's a very interesting finding. And then what do you want to see? In a minute, few minutes, they move something. So that's why now they want to check nucleus, and then definitely some such skeleton like actin, tubuling, or intermittent filament, they should be involved, right? Cell cannot move by themselves. Cell move their bone structure in human. The bone structure in human is actin, intermittent filament, or microtubule. So definitely actin is the most dynamic such skeleton. So they focus on the actin here. So here, first they check some ECM deformation. SG has a lot of deform. How you track this deformation? It's very similar data with this one. This is like without bead, they dye the gel and they track how they move. But here, to calculate the real, I can say, distance of the gel, and then they embedded the bead with which have process, and then you track the bead movement over time, and you can know their distance. So, for example, how we did this kind of study is that you made the fluorescence bead inside of gel, you encapsulate the cell together, okay, and then you capture that images 1 hour later, and then you add blebstatin to remove the ectomized contractility, and then this bead relapsed, move, because previously without blebstatin ectomized inhibitor, cell pull and push the ECM, right? So bead somehow deformed. But when you inhibit them so ectomycin contractility in cell, they go back because they don't have any power. So from the two difference of the bead images, you can calculate the distance. So this is the normal way, and then we can do this in that trend. So in 3D, they check the deformation. They deform a lot, like 1 micrometer. Maybe this is time scale of, sorry, time scale of 15 minutes. So 15 minutes is that you, maybe in this case, yeah, maybe you have seen the detail, but maybe, okay, after culturing, like right after adding, embedding the cell, and then, okay, after 1 hour later, 1 hour, and then 1 hour, and then 75 minutes, something like that. And then you can track two different process images. From the images, you can track distance like this. And then this rectification, and then they confirm, okay, they actually this cell scale is how much? Let's say this D scale bar is around 10 micrometer. This is 10, okay, 10, and then they move 1 micrometer, 10%. It's quite a lot, right? So this 10% are moving, but they're very small to detect, but we can know from this information. And definitely when they move the ECM, they need some cytoskeleton. So they check the actin first. Means so actin actin can be detected inside of all cell, even including nucleus. But here they highlight the cortical actin because cortical actin they should have a role to change the ECM, right? Because this is like cell is an interface. So that's why they call cortical actin. This is just normal affectin, but they are located in out near the cell membrane structure. So cortical actin presents over 5 minutes, but quite a lot in SG. Because of this, the cell can move the ECG. Without this actin, cannot move it. And then they check in detail. This is like 5 minutes. They check the 30 second, 0 and 30 second. Actually, this kind of live images you cannot do using normal cell. You have to transfer the cell using like live act plasmid, which has more actin. So, and then this actin has autofluorescence by themselves. So you transfer the cell with this live act plasmid, and then this active cell, they have, let's say, red fluorescence by the actin structure, and you can track it without staining. So for live imaging, the thing you have to transfer cell using certain plasmid, which has fluorescence actin. Then they track 0 and 30 second. Then you know this 30 second change over time, like this and that. And then they calculate the lost part from the beginning. This is similar, and then gained part. They combine them together as a dynamic area. Okay, so dynamic area as a percentage of total cell area is actually quite a lot, 5% always. But CG, they don't move them much. And then over 5 minutes, let's 5 minutes. So from this is 5 minutes, right? So from 5 minutes, how much they change? Average 5% here, but CG has less. So it's affecting dynamics, and then their cell movement also quite a lot in SG. So normally people show us like A and B is quite normal to show us. You can also easily imagine. You easily perform. Here, they highlight the time scale, minute to second. That's why they track the 30 second, very short time. And then from 30 second, they can detect the change. And then now they look at the nucleus. You can imagine this is a cell body, this is nucleus. Nucleus also they rotate, right? Cell when they move, definitely nucleus is move. When you are going, your brain is also going. Same thing. For CG, they didn't move that much. They track X and Y, they didn't move. But here they move a lot. Nuclear speed also from the center of this nucleus, you can track how they move it. Speed is higher. Nuclear track length. Speed is like during 5 minutes, how they move here to here, like this. Track is you can track it, like the length. So they track also, confirm again. And then this is like very obvious. When cell is moving, it's moving. But the very interesting point is that cell speed like this, but relatively nuclear speed is higher, right? So when you move like certain range, your body, but your brain move faster. So which means that they want to highlight this so dynamic movement by the nuclear. Even though they're moving 10%, but nucleus they move a lot, right? So this is like their speed and the angle also same. Angle is same because cell and nucleus they are stick together, right? So because that's why when cell is rotate, nucleus rotate. This is the meaning of this. So this is very accurate their quantification. So I want you to know this kind of thing. So when you see some change, you have to think about which kind of quantification you can do. Do your best. Reporting other people like this, you have to think about, I want you see some change. How can I quantify? We have image-based software, Fiji. So there's a lot of like software in inside, so you can track which kind of quantification you can do. And their correlation also a lot. The next figure 3 is about early stage cell tumbling. So we are highlighting from figure 1 a mini scale, right? 10 minutes, 15 minutes, and then during 1 minute, during 30 seconds, cell is moving, nucleus is moving. So they want to know, what's the meaning of this cell tumbling? This is really related to the cell's differentiation in chondrocyte, stem cell. Here they show us initially the chondrocyte differentiation, but the later figure, they show us another differentiation like bone or adipose tissue. So they want to know that this cell tumbling is essential for stem cell differentiation. Okay, which is that they're actually our figure 1 here. We show that cell differentiation in chondrocytes is very different, and then you observe cell is tumbling, right? Well, you can easily hypothesize that two should be correlated, right? So they want to regulate the cell tumbling here. So actually, they track all the time day zero. They're doing it right after adding the cell in the gel, same day, called day zero. And then they track right after embedding the gel, and then 48 hours later, 96 hours later, maybe during 1 hour, like that. And then CG, in supplementary, they also track 0 to 4 from CG. Speed is going down, but always higher than CG. Angular speed as well, right? And then they show us 0 for CG. This deformation also. Also, right? Says SG day 0 should be similar to these images, right? And then they top scale is 1 micrometer here, also 1 micrometer here. So you have to think, you have to also show like this. So when you use similar like tendency data, do not change your scale. Always you have to maintain the scale. And then people need to understand. Let's imagine here you use the scale like 2 micrometer to 0. Color can be changed, right? But someday this be sometimes this cell have 2 micrometer distance. But previous figure you show us 1 micrometer distance. But then they have same measurement. In that case, who can believe your data, right? You show us representative images, representative data, and then when you put the similar data in the figure, you have to show us similar pattern, right? And then they show us over time, this deformation is going down. Definitely, this all CDE and AB we previously observed, right? Speed displacement, affecting dynamics, nuclear speed going down. So this is very important. So when you show like figure 1 and 2 something, and then when you show another like over time, or when you add the inhibitor or activator, you try to track figure 1 to get the data set. Here, speed, angular speed, displacement, effect dynamics, mean speed, you have to show once again. Yeah, the whole things. Yeah. And then this is a good way how to. Then they check, oh, the cell tumbling is decreasing over time. So they want to control the cell tumbling by velocity, because as I said, actin cannot move by themselves. Myosin, normal myosin 2, should be involved. This is acting like a muscle in our body. So they treat blefastatin initially 1 day, 3 day, 1, 4, 3, 6, 7, 10 days, and they differentiate using chondrogenic media. But this initial 3 is like they inhibit the initial cell tumbling, right? And then this one is middle inhibit and late inhibiting. Doing that, in supplement, you can show them over time, maybe after 6 or 7 days, this cell tumbling is stopped. So that's why this point is important or not. They want to say that. So they track this control is without blefastatin treatment. This is CG. You can see more chondrogenic. But when they inhibit only 3 days, even 1, 4 days, they are severely going down. But 3, 6, 7, 10 days, not that much. So they highlight this initial cell tumbling is very important for fundamental differentiation. So this is very logical. I'll show the data. So we have figure 6 today. So I'm gonna stop here. So I want you to know this kind of very live imaging, even in second or time minute scale. And then how you show the data, quantification. Actually, let's imagine for making gel, maybe it takes like 1 or 2 months. Okay. And then let's start the experiment. 10 minutes is finished, right? and then second finish like that. But maybe you need more time to quantify. So okay, you can do experiments like 1 hour, but you can quantify like day or 1 week to show us something. Yeah, there is a good way how the good people are doing in mechanical battlefield. You try to quantify something, right? And then you can follow other people's how they do, and then you can also think about what is new like quantification by your hand. This is one of like creativity idea how you upgrade your paper. Okay, any question? Okay, okay. See you next week, Tuesday.